Neuroprotection is the mechanism by which the central nervous system of our body prevents or delays neural degradation or injury caused by reactive oxygen species or other endogenous factors that can eventually result in cell death or long-term diseases like stroke and Alzheimer's. Another important mechanism that helps the body from the severe side effects of inflammation or swelling induced as a response to pathogenic factors is anti-inflammation. There have been many studies that have showed that vanillin, a compound derived from vanilla beans, which has widely been used in flavoring agents, has both neuroprotective as well as anti-inflammatory effects on human cells, which is what was studied in this case. Firstly, Soxhlet extraction was performed to obtain vanillin from cured vanilla beans, and the extract was analyzed using gas chromatography to quantify the amount of vanillin present. In order to study the neuroprotective effects of vanillin, initially a neurotoxic model of hydrogen peroxide was developed by testing doses of H2O2 ranging from 10 to 1000 micromolars on Scheiwei cells for 2 hours. An MTT assay was performed on these cells to measure the percentage of the cell viability, and a cytotoxic dose was chosen based on the results and used in another neuroprotection assay, where neuronal cells were plated and pre-treated with vanillin doses ranging from 5 to 500 micromolars for 24 hours. An oxidative stress was then induced on these cells using the cytotoxic dose of H2O2, which was decided to be 300 micromolars, and this was left on for 3 hours before an XDT assay was performed on them, again to measure the percentage of cell viability. To study the anti-inflammatory effects of vanillin, THP1 cells that were differentiated into macrophages were pre-treated with vanillin doses for 24 hours, after which inflammation was induced using lipopolysaccharides, or LPS, a component of bacterial membranes. This was left on for another 24 hours, after which RT-qPCR was performed on these samples, and the expression of the CXCL12 gene was measured relative to that of the GAPDH gene, which was the housekeeping gene. The CXCL12 gene is responsible for coding for a pro-inflammatory cytokine that synergizes with other cytokines and promotes overall inflammation. Although the extraction technique worked, the concentration of vanillin was too low to be used as a treatment dose, and so a vanillin standard was used for further experimentation. The H2O2 neurotoxic model showed that all the tested doses were able to reduce the percentage of cell viability compared to the control, as you can see in figure 1a. And from the neuroprotection assay, it was found that all the doses of vanillin that were tested protected the cells from any form of degradation, and this can be seen in figure 1b. This is because the high free radical scavenging activity of vanillin allowed it to interfere in the oxidative chain reaction by providing the peroxide ion a proton and neutralizing it, preventing it from causing any cellular damage. The PCR showed that there was a downregulation of the CXCL12 gene in the samples that were treated with 500 micromolars of vanillin, as observed in figure 2. This suggested that the gene could not code for its cytokine, which was the stromal-derived factor 1, causing a disruption in the inflammatory signaling cascade. Thus, from these findings, it was concluded that vanillin indeed had both neuroprotective and anti-inflammatory properties, which are beneficial to the human body. If the study was to be continued by another individual, I would suggest studying the toxicity of vanillin alone to see if it had any effects on the cell viability prior to subjecting the cells to any form of oxidative stress. Furthermore, a wider range of doses of vanillin can be studied to identify the dose at which downregulation of the gene begins.